Hello, welcome to the Catana tutorial video. Catana is a web application for visualization and modeling of DNA protein nanostructures. As you can see on the main page, it enables you to visualize, model, assemble or predict various structures. In this video, we will provide you with an overview of the application, introducing you to the user interface, some design concepts and general ideas behind the Catana. So let's start with the user interface. So the top part of the Catana is a navigation bar which allows you to load or export structures, run some dynamics procedures, change some settings of the scene or camera, change layouts of the user interface, or access help, user manual, or some additional features. The left bar of Catana is a controls panel which allows you to create DNA single strands, double strands, modify proteins, and other structures. The right bar of Catana consists of two parts. The top part is workspace, which shows you which structures are loaded into the Catana. So as you can see, currently we have all atom tau protein loaded and coarse-grained DNA. Then the bottom part of the right bar is inspector, which shows details of currently selected structure. So you can see if you click in the workspace or if you click on the structure in the scene, the inspector shows details and some options or features related to the structure. The bottom bar of Catana shows the version, currently 1.1, and some scene settings, allowing you to show the gizmos at the center of the world or the origin, or to show the modeling grid, for example. Finally, the top right part of the top bar shows some options for enabling gizmos for rotation or translation of structures, together with possibility to choose if these gizmos will behave in a particular way. So this is for the overview of the user interface. And now let's get to some basics. So for example, let's say we want to load the PDB structure. So first I will hide these two prepared structures and then head to structure import. So let's say we will load this one BNA, which is double-stranded DNA PDB, and click on the download and import. And now you can see the DNA was loaded into the scene. What you can also see that in the workspace, now we have three structures. Two of them are grayed out because they are hidden, and one is, let's say, fully visible because it's the one BNA we just loaded. So what we can see that if we click on this one BNA, we can see some options in the inspector, which allow us to duplicate the structure, delete it, center on it, and so on. We can see also various representations, sequence, some placement details, allowing us to move the structure without the gizmos, and some details about the structure, listing the number of polymer chains, allowing us to change the assemblies, and so on. So let's start, for example, with the representations. So as you can see, we have currently three representations active. The ball and stick, which shows only ligands, which are not present in one DNA structure, so we can, for example, delete this representation. Base representation, which emphasizes DNA bases, so if we hide it, we can see the DNA bases will be shown or hidden, and the cartoon representation, which currently shows the DNA backbone. We can filter out this representation, so we can, for example, show only base for guanine bases, and we can also use color filtering, which will emphasize, for example, only the guanines in yellow color. Similarly, we can again hide these color filter strings, and we can also, for example, change the color scheme to render the cartoon representation with colors based on the residue names. Or, for example, we can again use this color filtering to see that, let's say we have residue with index 5, over there residue with index 9. So we can make nucleotides with residue indices 5 to 9, for example, again yellow, and emphasize them. So these are some of the representation settings. Then we have, for example, the action settings, which allow us as I mentioned, some features like duplication, deletion of the structure. So let's delete this one DNA, and we can see that it disappeared from the scene. So let's show again our pre-prepared structure, and let's see. So in the workspace, you can see that we have this small icon which shows if this structure is all atom structure, as in the case of this all atom tau protein, or coarse-grained structure. These icons, or basically the Catana supports basically both all atom and coarse-grained structures, and there are different tools you can use with these two paradigms. So if you head to the layout, you can see that we have coarse-grained modeling layout, which offers some features which allow us to create or design coarse-grained structures. And then you have all atom modeling layout, which allow us to modify all atom structures. So if you want to work with the respective or with the corresponding structure, you have to make sure that you are in the correct layout. So this is some of the basics, and let's now perform some very basic operations to see basically how you can use Catana. So let's say we have this nicely positioned DNA, but we want for some reason to move it out and take a look. So we can select this translation gizmo, click on the DNA structure, and move it away from the protein part. Other option is that you hover over the residue or nucleotide, or let's say element you want to translate, and you just click your left mouse button and drag. And now you can move the structure in the scene, freely in the scene. The same applies for rotation. So if you click over there and drag, then you can rotate around that point in the world. Or you can simply click and release the mouse button and show the gizmo, and then rotate using the gizmo. As for the translations and rotations, we have this option which allows us basically to change the orientation of the gizmos to be aligned with basically with the principal or global or local axis of the component. In this case, the alignment is nice by default, but in some cases this modifies basically the behavior of the gizmos. So for example, let us go to the coarse-grained modeling and select the create double strand tool. Now I will hold left mouse button and drag and create 85 base pairs long double strand. So as you can see, this is coarse-grained DNA double strand with some random sequence which is generated by default. And if I want to to make it easier, I will show also the other DNA so we can see the effect of the translation. So if I want to translate the structure now, I can click on some part of it and move it around. As you can see, if you have the world gizmo, the axes are rotated aligned with the world axis of the Catana stage or Catana scene. But if we show for the principal axis, the gizmo is aligned with the structural details. So for example, in the case of DNA, it allows for much easier positioning because the z-axis is aligned actually with the helical axis of the DNA. So this is for some translation options. And now we can also show that the Catana allows to transform between the coarse-grained and all atom worlds. For this example, you have this convert to all atom function in this coarse-grained structure. So if you press on it, the Catana will generate atoms for the DNA. So let us show the ball and stick representation and perceive that our DNA is now all atom structure with all the atoms generated. Similarly, if you want to go from all atom structure to coarse-grained, we can click on the structure, on its inspector, and see convert to coarse-grained. And this will duplicate, or this will make the structure again coarse-grained. So these are some of the features of Catana. So again, we can show some positioning, so we can drag and drop and move along the axis or move freely. So if we want to somehow again position the DNA inside the tau protein, we can simply move it, rotate the scene, and just position it to just give us some perspective how it may look. Catana offers some additional features, so the import can be from the RCSB database, AlphaFold protein structure database, or some custom website, or you can drag and drop some local file. So for example, we have some plain UNF file, so we can load it, wait a moment, and see that the structure has been loaded into Catana. So this is a coarse-grained structure of a plane. So additional things are offering basically export of structure, so we can export structures which are loaded in Catana into various formats, into PDB, UNF, as a FASTA file, or as a screenshot. So for example, for a screenshot, we can again position these structures in some, let's say, close proximity. We will zoom onto the structures, go to the export, screenshot, and click on preview. And we can enable some settings, for example, upscaling, transparent background, or trimming of the space. And this will generate nice transparent screenshot of the structures in the scene. There are also some other scene settings, so we can preview the structures on the white background, or we can modify the fog properties, so it's, let's say, more aggressive, so it's more obvious that there is some fog happening. Then we can toggle the layouts. For example, the visualization layout hides the left and right bars, so you can perceive the structures without, let's say, with maximum visible space. And then finally we have also the scripting layout, which enables to execute some commands. So for example, if we run this command, it will create a coarse-grained single-stranded DNA with a sequence Agata. A similar feature or similar thing can be achieved with this structure create menu, where you can create coarse-grained DNA or peptides based on the sequence. So you can see again we have created single-stranded sequence, single-stranded DNA, or we can create single-stranded, oh no, not single-stranded, but we can create a peptide, simple one. As you can see, for coarse-grained models we have different representations. Then for all of the modeling ones, so we can use, for example, this tube representation to see this very small alpha helix-like structure. Catana also offers possibility to predict structures, for example, using the AlphaFold predictor, or run some relaxations or some dynamic simulations, which might be explained in other videos. So this is the basics of Catana, and we hope you will like the software. And if you have any questions, feel free to ask under the video or on the main Catana welcome page, which contains email to us. So thank you for watching this video and have a nice day.